finish up with a discussion of protein post-translational modifications. So what is a post-translational modification? It's a change made to the protein after translation. So remembering we've got our DNA gets transcribed into RNA, mRNA, the mRNA gets translated into protein, and then we're talking about a covalent modification that happens after this protein, or at least that amino acid in the protein, has been translated. So proteins can have a complicated combination of different post-translational modifications. Don't get the idea that a protein can only have one specific post-translational modification. It just depends on the amino acids in the protein. And then even though post-translational modifications, or PTMs, you'll sometimes hear them called, even though they're covalent modifications, they are often reversed by an enzyme. So these are dynamically going on and off in the cell all of the time. So post-translational modifications, again, are covalent modifications to the protein structure. Here are several post-translational modifications. So for example, we can add a sulfate group to a tyrosine, we can add an acetylation to a lysine, we can add a methylation to a lysine. There's a lot of these, and they're all important. The one I'm going to focus on in this class and hold you responsible for is phosphorylation. So that's when we have this phosphate group, and we'll talk more about this, but there are specific amino acids that you can modify by phosphorylation. Outside of those small chemical changes, we can also take a protein and post-translationally modify it with a whole protein. So one example is ubiquitin, is the name of a whole protein. We can take that whole ubiquitin and covalently attach it to the protein we're interested in. The cell does this all the time. A prenyl group is a type of lipid. We can take a lipid and covalently attach that to the protein. We can take a glycosylation, which is a carbohydrate, and covalently attach that to the protein. And then, of course, our small chemical groups. So we're going to step through a couple examples of each of these. Starting with phosphorylation. So phosphorylation can occur on serine, tyrosine, and threonine, where that side chain hydroxyl can form a phosphate ester whenever a phosphate is transferred over from an ATP molecule. So for example, here's our serine side chain and a hydroxyl. We can have that changed into a phosphorylated serine, so there's our phosphate ester. Here's our tyrosine, we've got the same thing, and here's our threonine, we've got the same thing on that hydroxyl. So that's one thing I want you to be able to do in this class, is be able to draw phosphorylated serine, tyrosine, and threonine residues. As I said, this phosphorylation is reversible, so there are enzymes going around the cell that are dynamically adding and removing phosphorylation to certain proteins. Here's one example. So there's a cell signaling pathway where we've got like a G protein coupled receptor. It activates this protein called RAS, which activates this protein called RAF, which phosphorylates this protein called MEK. So now we've got phosphorylated MEK, which is activated. MEK goes on to phosphorylate this other protein called ERK, so we've got phosphorylated ERK, which is activated. So what you're seeing is there's this whole downstream cell signaling cascade where phosphorylation is the key on-off signal for whether this signaling cascade is on or off. So again, notice this protein ERK in this pathway. So if we take a closer look at ERK, we have the structure of this protein with no phosphorylation, what you see here, specifically this group here and here, with phosphorylation at a threonine and a tyrosine, so you can see the phosphate groups there and there. And then if I overlay those two structures, so gold is with no phosphorylation and red is with phosphorylation, we can see there's some pretty big differences in the protein structure. So one of the reasons could be that a phosphorylated tyrosine is going to form really different intermolecular interactions than a regular tyrosine, right? We have a charge versus not. So that can have big implications for the way this protein is folded. All right, so that was phosphorylation. Let's take a look at another example, how we can have lipids as a post-translational modification. So a palmitoyl, that's this group right here. So we can have palmitoyl attached to an enzyme, and then a certain protein that has a cysteine residue on it. And then we have enzymes that add or remove this palmitoyl group from this protein. So the product could be a protein with this palmitoylation on it. So what does that mean? Let's say our protein is this anchoring G, just for example. When it has that palmitoyl added onto it, the protein stays anchored to the membrane because the palmitoyl, that long hydrocarbon chain, embeds into the membrane. Whereas if we have an enzyme reverse that, cut that palmitoyl group off, now anchorin G could unbind the membrane and float around free. So the cell dynamically controls membrane binding through this type of post-translational modification. Here's another example. I talked about how we can add and remove whole proteins as part of our post-translational modifications. So one example is the protein ubiquitin. This is ubiquitin. So you can see it's a whole protein with its own three-dimensional structure and stable fold. It's abbreviated capital U, lowercase b. So here's our protein of interest. If the right enzymes create a long chain of polyubiquitin, many ubiquitins bound, it signals the cell to go and degrade this protein. It says, this is trash, I don't need it anymore. So it sends it to this larger structure called the proteasome, which is also another protein. It feeds it through, cuts it up, and spits it out as free amino acids or peptides, which can then be recycled and used for building new proteins. So that's one example where we can use a ubiquitin post-translational modification to signal something to the cell, in this case, protein degradation. All right, so practice problem. Draw a phosphorylated tyrosine. As I said, that's one PTM I want you to be able to draw. How does this covalent modification change the tyrosine's propensity to form favorable interactions with the lysine residue?